Our next speaker is Eleanor Kennedy. And Eleanor is actually a scientist and has come all the way from London. Was trained in Glasgow and did a postgraduate training after that in Leicester, and has done a lot of work with us in diabetes. At the moment, he's a group leader for University of Geneva in Switzerland and has had an MBA, as if she hasn't got enough to do to top it all up. And I refer you, actually, before Eleanor can actually talk, in your booklet, there is a AASD e-learning website on page 43. So you'll see some excellent work that Eleanor has done to educate us all, or to keep us well educated in diabetes. And there's more modules about e-learning that are coming through in the future. So Eleanor is going to talk to us about getting involved in diabetes research. Thank you, Eleanor.
Thank you, Mick. Thank you very much for that lovely introduction, and thank you for being here. Thank you to the organisers for inviting me to come here. We've had an awful lot of very interesting talks this morning. We've gone from artificial intelligence to diabetic neuropathy to diet. And the one thing that holds all of those together is research. And that's how I'm going to finish this session, talking about getting involved in diabetes research. And I don't know, some of you may have been at my workshop yesterday about getting involved and how to find funding for research. This one? No. This one? No. How do I get to the next one? Which button am I pressing here? Oh, there we go. The first thing I will say is just a small disclaimer. I've been upgraded. I've had a free upgrade for the duration of the conference. I've been put in as a professor. I am a mere doctor. From a research point of view, as healthcare providers, you should be really pleased where research has taken us. It's taken us here from 1921 with two researchers, Banting and Best, along with their colleagues Collip and MacLeod, discovered insulin. To 2018, less than 100 years later, and the diabetes armamentarium is really quite full. We've got DPP-4 inhibitors, we've got SGLT2 inhibitors, we've got GLP-1 receptor agonists. And all of that comes to you as healthcare providers through research. And that research process is extremely long and has become a lot longer thanks to the FDA and EMEA's cardiovascular outcome trial guidance. But we have to start with an idea. And that's what we did in the workshop yesterday. We need to have novel ideas to get new research. We then need to go through the basic research, which is where I sit, looking at animal models, looking at cell models. And by the time you get through to clinical trials, you get through your phase 1 safety trials all the way through regulatory approval and post-marketing trials, that can take 10 to 15 years, as you know, you then get out into patient care. And that's really good. But the problem is that if you don't have your patients in those research trials, we don't actually know if those drugs are going to be effective. So if you look at the EMPA-REG trial on empagliflozin, which many of you will be familiar with, 42 countries, but none of them in the Middle East and North Africa. If we look at the CANVAS trial, which was just reported last year on canagliflozin, lots of different countries, but nothing in the Middle East, nothing in North Africa. And if we look at the LEADER trial, Novo's liraglutide study, you can see that there was one site in United Arab Emirates that recruited 31 patients. The problem with that is that if your patients aren't involved in clinical trial, we don't know that those drugs are going to work. We don't know that those interventions are going to work. And when we look at this region in the world of research, and here I've filtered for medicine, which may not be the best filter, but it's how I filtered it. You will see that from a medical point of view, the countries that you might expect that would be publishing the most are America and the UK and Germany and so on. The nearest country in this region that gets into the list is at number 24 and is Iran. And our lovely host country at the moment limps in at number 39. And what about trials? So that's all of medical research. But what about trials? Now, this is tiny little font. I don't expect those of you in the back to be able to read this. But if we just look here at North Africa and the Middle East, you can see, and if you look at the very right-hand column, 14.6 trials per 10 million of population. Now, when you put that into context with where I'm from, which is Western Europe, we dwarf that logarithmically, and we are dwarfed still further by the States, which means that clinical trials are actually dominated by people like me, and not like people that are in your patient base. But you are getting better. This is in 2016. You can see that about 9,500 studies were being done in the Middle East region. Unfortunately, this is dominated by Turkey. Turkey are running away with clinical trials at the moment. 2 years later, that's improved. You're at over 11,500. But again, Turkey is taking the lion's share of that. So what are the possible barriers? Why are you not getting involved in research? There are probably more than this. I've gone with these 4. There's the regulatory firewall. And these are problems for all countries. It's not just problems for this region. Regulatory firewalls are about the monitoring and adhering to regulatory change. There is a big issue here because some of the countries in this region, if you are trying to import an investigational new drug, an IND, it can take between 2 and 3 months, 60 to 90 days. Now that has massive ramifications for how and when you can start any clinical trial. But the establishment of clinical research organisations throughout the region locally, I think will help. I think that will really help to move that regulatory bottleneck. And then, of course, there's oversight. And oversight is about good clinical practice, it's about standard operating procedures, and a lot of your organisations and institutions will already have all of this in place. There is a big time and big financial investment in trial monitoring, but once the training of the staff has been done, the staff are then trained, and they can then work on any trial. So it is a very important oversight. It's incredibly important for the sponsor, but once it's in place, it's in place. And then, of course, there's informed consent, and that it really is quite a big issue, because the FDA in the States and the EMEA in Europe are naturally and very obviously very, very keen on informed consent. All study materials have to be properly translated and accurately translated. And there are issues with that, because a number of languages, if you take a principal investigator and you translate investigator, the closest word becomes interrogator. And actually, that's a very different thing. An investigator is non-threatening, but an interrogator is very threatening. So there is a possibility of misinterpretation, and accurate translation does, of course, have its own challenges. And then, of course, there's this region, medical schools and teaching hospitals. You've got a really rich tradition in science and innovation. There are possible paucities, perhaps, of teaching research methodology in postgraduate training, perhaps a lack of willingness to encourage patients to become involved in trials. Importantly, lack of protected time for being involved in research. That's always a big one. That's a big one in every country. And perhaps a perceived lack of reward, although I would argue that being involved in any kind of research is reward enough. So why do we need clinical trials? Well, of course we need them. We need to demonstrate safety. We need to demonstrate efficacy. But do we actually need the big randomized controlled trials? They're still viewed as a gold standard. But I'm going to take you back about 250 years and introduce smallpox to a diabetes and metabolism conference, which seems a little left field. But Edward Jenner asked the question, why do milkmaids never get smallpox? Smallpox, as you know, is a highly contagious viral condition caught from cows. Milkmaids historically were the women who went to the fields and the sheds to milk cows manually by hand. But they never got smallpox. So Edward Jenner recognised this, and he said, well, that's quite interesting. Why do milkmaids not get smallpox? So he took a cowpox blister from someone who was affected with smallpox, and he scratched the inside of it out and then scratched it into the skin of an 8-year-old child. And the boy did not get smallpox. And the following week or 2 weeks later, he did the same thing, and no blisters appeared at all. And the vaccine was a success. And Jenner called this new method vaccination. And every time you recommend a vaccination for somebody, you are actually harking back to one of the very first clinical trials that was ever done, because vacca is the Latin for cow. But Edward Jenner was absolutely ridiculed for this. He did this one study, and he couldn't publish it. None of the publishing houses in the UK would look at it. And he was roundly abused in the British media, and a very famous cartoon, which you can see there. And it wasn't until after 30 years after his death, that smallpox vaccination was made compulsory in England and Wales. So why is that important? That's important because what Jenner did was an N of 1 study, and he also got parental consent. So what is an N of 1 study? An N of 1 study is the sort of thing that you could be doing. You could be doing every day in your practice. They are single subject, randomized, crossover studies, which are done in collaboration with patients, very much harking back to the AASD consensus statement, where you're working with patients to get management plans written up and agreed upon. They offer an alternative to large randomized clinical trials. They're much cheaper to do. But if you are running an N of 1 trial, and you are running an N of 1 trial, and you are running an N of 1 trial, what you then get is lots of N of 1 trials which merge together to give real-world evidence. And in populations that have not been exposed to the big randomized clinical trials, this is a really good way to get the evidence, to get the research to back up the use of certain drugs or certain treatments. And he also got consent. He also got parental consent. He asked the boy's father, would it be okay if he inoculated his son with some cowpox, and the father strangely agreed to it. But he did get parental consent. So as a trial, Edward Jenner's clinical trial, I think, was a really interesting one. And as I said, now that we're in a new era with new ADA, EASD guidelines, where we're putting the patient really at the centre of care, by working with the patient in N of 1 trials, you have the opportunity to start putting together not just robust management plans, but robust N of 1 trials. And you might say, well, how can a small trial actually work in diabetes? Surely you need these massive EMPA-REG, UK PDS, DCCT trials. And actually you don't. And this is a diet-based piece of research following on from Tanya's wonderful talk on diet and diabetes. This was a study that was done by Roy Taylor's group in Newcastle in North England. And this garnered a huge amount of media coverage. He did a small study that was wholly funded by Diabetes UK, about £300,000, a very small amount of money to do this study. And all he did was recruit 11 people. And he didn't even control it properly. He only had 8 people in the control arm of this. But what he did was he put these 11 people on a very draconian 600 kilocalorie diet a day. And he showed that within 1 week, fasting plasma glucose was normalized, insulin suppression of hepatic glucose output was improved, and first phase insulin response was also increased, leading him to say that abnormalities underlying type 2 diabetes are reversible by reducing dietary intake. And that spawned this, and this was announced at the IDF conference at the end of last year. He leveraged £500,000 out of the same organisation to run a huge trial which showed that essentially you can put type 2 diabetes into remission. You cannot cure it, but you can put it into remission on a very low-calorie diet. And this one study that came from just 11 people according to Web of Science, just yesterday, it's a hot paper, a so-called hot paper, and if the citations continue at the same rate, it will be one of the top 0.1% of papers published in the field of clinical medicine. So yes, small studies really do matter because they can lead to something much, much bigger. So what are your possible opportunities to get involved? And again, there are probably more than that. But if you look at the disease pool, I'm not specifically talking about diabetes, but if you look at the disease pool, you have genetic and cultural differences within the region that offer really unique opportunities, I think, for research. You've got different ethnicities. Historically, there has been a higher rate of consanguineous marriages, and with consanguineous marriages, you tend to get mutations or certainly sequence changes within certain genes that then crystallize and become more apparent in later generations. And geographic isolation. And you do have certain disorders, certain genetic disorders that are much more common in this region, certain hemoglobinopathies. You've got glucose-6-phosphate dehydrogenase deficiency, which leads to hemolysis. You've got a whole raft of congenital malformations and several metabolic disorders that I'm not going to go into. But as they are mostly autosomal recessive, which would hark back to a consanguineous derivation. But if you look at just the genes that are implicated in type 1 diabetes, there's lots of them. There's loads and loads of them. But all we've done is look at those genes in the same populations that are involved in the big clinical trials. They're not genetic samples from this region. When we look at type 2 diabetes, there are even more. And I know from various travels that I've had around the Middle East that you have samples. There are samples in freezers all over the place. You might be sitting on some of the biggest genetic breakthroughs in type 1 and type 2 diabetes, but we don't have access to those samples, but you do. And what about recruitment? You have population characteristics, you've got a growing healthcare infrastructure, you've got state-of-the-art communication that make it actually a very ripe place to do research. And of course you've got cost. Cost of trials, as I said, with the cardiovascular outcome trial, trials that are demanded by FDA and EMEA, are driving the cost of trials up. So a lot of the pharmaceutical companies and biotech companies and device companies are looking to do trials outside of the US and Western Europe because we're just getting too expensive. Here, you could make significant savings. And of course, you are an emerging market, and you're an emerging market that is actually outstripping the places we thought were emerging markets, Brazil, Russia, India, and China. You have a growth rate, projected growth rate, of about 10% in the pharmaceutical arena. So what have I managed to tell you? I've had to race through this because I'm critically aware that after 20 minutes, these boys are going to switch off my slides. What I've tried to show you, I hope I've tried to convince you, is that in the last 100 years, diabetes research has moved on in leaps and bounds. We have gone from having really just insulin to having a really quite full drug armamentarium. But we only have that because of research. We only have that because of research that has come from a very basic molecular background through clinical trials and into a patient base. The vast majority of these trials have been done through large and very expensive multinational randomized control trials. But actually, I feel that now the tide may be turning on the big RCTs, and we may have much better opportunities to get real-world evidence that is both compelling and is done in smaller, more targeted studies in regions that historically, like this region, have not been involved with, have not had access to be involved in the large RCTs. And I think that this region is really ripe for research. I think it's ripe for interventions that are done on a clinic-by-clinic basis or a patient-by-patient basis on small N-of-1 trials that can, added together, can make really quite compelling real-world evidence. But you can also exploit the new omics revolutions, and you can exploit the world of genetics research, because I think that as an area, this is an area where you could really make enormous impact on an international stage. And of course, I will finish there, and as always, I will say, if you have been, thank you very much for listening.